Good afternoon and welcome to the webinar. I'm Dr Alison Foster and I'm the Head of Technical for Preclinical for Quay Pharmaceuticals, a CDMO based in North Wales in the UK. I'm joined this afternoon by my colleague Dr Mike Frodsham, who is our Chief Technical Officer, and we will be talking about lipid-based drug delivery from preclinical to first-in-human studies. First, a little bit about Quay Pharma. We're a contract development and manufacturing organisation based in the UK just outside of Chester. We are a family-owned business. We were founded in 2002 by Mike Rubinstein, who was a professor of pharmaceutics and chemistry at Liverpool John Moores University. We've now got close to 200 people on site, and we're experts in formulation of molecules for systemic and localised delivery. We consider ourselves specialist formulators of problem molecules, including small molecules, live biotherapeutics, and antibodies and other large molecules. And we can perform GMP clinical manufacture of all the products we develop. I'm going to focus firstly on developing formulations for preclinical studies. Firstly, the main question is why we would choose lipidic delivery. If we consider molecules that fall into class 2 of the biopharmaceutics classification system, so those are molecules that are poorly soluble but highly permeable, then there are a variety of methods that are typically used to improve absorption. For those molecules that fall into class 2B of the developability classification system, which was defined by Jennifer Dresman as molecules that have solubility limited absorption, then there are two main options that are generally available to improve bioavailability. These are lipid formulations and amorphous solid dispersions, shown on the right. A lipid formulation route is selected if the molecule has a low water affinity with a high log P, whereas molecules that have a high melting point and medium log P would typically be formulated as an amorphous solid dispersion. So what are the benefits of lipid delivery? It is possible to solubilize and enhance the bioavailability of a poorly soluble drug by maintaining the solubilized state until the site of absorption is reached. Lipid formulations can mimic a positive food effect without the need for food. They can help to decrease the first pass metabolism of the drug by recruitment of intestinal lymphatic processes. They can increase the permeability of the drug through inhibition of the P-glycoprotein efflux transporter mechanism. Dose range finding studies can be performed easily by dosing increased amounts of the formulation. And finally, development time can be reduced as a formulation can be dosed via oral gavage in preclinical studies, and small quantities are easily manufactured for early clinical trials, and scale-up is straightforward for commercial supply. The lipid classification system is a good reference tool and can aid formulation design. Futon and Porter define 5 types of lipid formulations depending on whether the formulation is more oil-based and requires digestion, which is type 1, if it is a self-emulsifying system, which is a type 2 or 3, or if the formulation is actually lipid-free, indigestible and forms micelles in solution, which is type 4. This classification system gives example compositions for each formulation type, expected dispersion in aqueous or biorelevant media, and whether the formulation class is digestible. As we move towards type 4, then there is an increasing risk of reduction in solvent capacity on dispersion and a greater chance of precipitation of the drug, unless precipitation inhibitors are added to the formulation. So how do we design a lipid-based formulation when we have 5 classes to choose from? So I'm going to take you through this flow diagram on this slide. The first step is to perform a solvent screen of the API in a number of individual lipids. We usually aim for about 20 different options at this stage. If the entire dose can be solubilized in a single lipid, and this can be for a preclinical study as well as a first-in-human study, then the simple system should be tested for dispersion or precipitation in biorelevant media. Provided a dispersion or micellar solution is formed, then that's an easy win. If, however, and usually this is what normally happens, the entire dose can't be solubilized, then we need to consider excipient mixtures based on the results from the original solubility screen. In parallel to this, the excipients that we choose should be miscible with each other at the desired concentrations, and we can target the suggested compositions from the lipid classification system as a guide. Clearly, previous experience can also help guide excipients or formulation selection at this point, and often companies such as ASSAY and Abitec will also share phase diagrams that they have generated, so a lot of the hard work has already been done. Once the selection of a suitable miscible excipient combination has been identified, rather than wasting precious API at this point, the placebo formulations can be tested for dispersion and droplet size can be measured with dynamic light scattering. Provided a suitable dispersion is achieved, then the API can be introduced to determine if the target dose can be achieved. If so, then a precipitation or dispersion test in biorelevant media can be performed, and if a dispersion is formed, then this lipid formulation can be taken forward. An example of how a rapid initial solubility screen can be performed is shown in this video. API can be weighed into individual vials, or, if there is limited API available, then a solution of it in a volatile solvent can be dosed in a microtiter plate, evaporated, and then the lipid or solvent can be added to the dried API, and that's what's shown in this video. In this case, less than 1 mg of API can easily be tested for solubility, provided it has a UV chromophore with an absorption of more than 300 nanometers, and this can even be tested for assay using a plate reader to save having to develop an analytical method at this stage. However, particularly with lipid formulations, it is best to have a draft assay method that we can use for solubility determination, as often the lambda max of the API coincides with the absorption from the lipids. In terms of analytical method development, there are some key points to bear in mind. The diluent needs to be miscible with the lipophilic excipients, so an organic modifier will need to be added, such as THF. Use of a guard column can help to protect the column from the excipients as well. System suitability data should be monitored to detect any early potential issue relating to the column. A precipitation or dispersion test is an important tool during the development of a lipid-based formulation. The formulation should be tested for precipitation of the API upon dilution with biorelevant media. If the A precipitates, then surfactants or precipitation inhibitors can be added if a backup formulation is not forthcoming. Dynamic light scattering can confirm droplet size and stability of the resulting dispersion for type 2 or type 3 formulations. In the next few slides, I will present a case study that we worked on recently, where we were asked to prepare a formulation for a 1-month tox study. In this study, the API was to be dosed via oral gavage in rat and monkey up to 1000 mg per kg, which thus required the formulation to be at a concentration of 200 mg per gram. Minimal API was available, less than 6 grams, and a fast turnaround was required as the annual study had already been arranged. The API had a medium log P, very poor aqueous solubility, was amorphous and non-ionizable, and had previously been shown to have good solubility in semi-polar solvents, surfactants, and oily vehicles and excipients with a medium HLB. The table shows the results from the in vivo PK, where the formulation was dosed in DMSO, which is obviously not suitable for the tox studies. The aim of the project was to generate a formulation that would achieve the required exposure without using DMSO, and to generate a formulation that was suitable for high dosages. Here I have summarised the timeline that we were bound by and the API quantities that we used for each stage. First of all, we developed an assay and related substances method by transferring in a draft HPLC method for the API. This took approximately 2 weeks and used around 200 mg of API. We then performed a solubility screen and determined the solubility used in the assay method. In total, we screened 16 excipients at this point using 500 mg of API with a turnaround of 1 week. Following the results of the solubility screen, we then looked at possible formulation options, generated 6 prototypes that were tested for precipitation in biorelevant media. This stage used approximately 1.5 grams of API and took 1.5 weeks. Finally, we scaled the 2 most promising formulations so that we could put them on a 10-day stability trial, testing after 5 and 10 days for assay and related substances, and to check that the formulation still dispersed successfully. This final stage used 1.8 grams of API and took 3 weeks. Analytical method development. We transferred the HPLC method provided to UPLC, which used a shorter column and smaller particle size in order to reduce the runtime. This was due to the number of solvents that we planned to screen later on. The resolution was evaluated using the API and the 2 impurities that were provided by our client. We performed a filter study, evaluated the solution stability, and assessed the specificity using the excipients planned for the solubility study. Linearity and sensitivity were assessed at both the assay and related substances levels. For the solubility study, 16 excipients were tested using minimal API. We targeted a concentration of 100 mg per gram at this point. However, we found that several of the excipients that we had selected achieved this target concentration, so we performed an additional visual assessment using selected excipients targeting a higher concentration of 200 mg per gram. From these results, we selected lead options for formulation development. For the formulation development, we prepared the 6 prototypes which are listed in the table. These formulations fell into type 2 through to type 4 of the lipid classification system. Following a visual precipitation test, formulations 2 and 6 both formed a precipitate, and it also appeared that a precipitated formed with formulation 4, whereas for formulations 1, 3 and 5, these all gave reasonable dispersions with no precipitate. It was agreed with the client to progress formulations 3 and 5 as the high lab result content in formulation 1 may cause some issues when dosing into animals. The stability of the 2 lead formulations proved acceptable over the 10-day time period. It was noted that the colour of the formulations deepened slightly, but this was particularly true for formulation 3. However, there was no change in assay and no significant change in related substances over this time period, as you can see from the data in the table. This slide shows the DLS results of the dispersion for formulation 3 in FASTGIF and FASIF. For this formulation, a slightly smaller droplet size of around 300 nanometers was obtained in FASTGIF and around 800 nanometers in FASIF at the initial time point. However, the DLS results appeared different for the older 10-day sample, or there was no difference visually. Similarly, this slide shows the DLS results of the dispersion formed when formulation 5 was added to FASTGIF and FASIF. In this case, the dispersion showed a less uniform droplet size throughout the test, as you can see from the data in this slide. So a few conclusions following the preclinical formulation work. Formulations 3 and 5 were successfully scaled to 4 grams. There was no change in assay or any significant change in related substances over the 10 days. The dispersion test showed an increase in droplet size with older samples, but formulation 3 generally produced a better dispersion, and so this formulation was selected to progress to the tox study. Finally, the toxicokinetic results. A dose range finding study in rats indicated that exposure increased in a dose proportional manner with increasing doses from 30 mg per kg to 300 mg per kg. Exposure in rats was lower than achieved with the original DMSO containing formulation, but was sufficient to achieve the objectives of the tox studies. A dose range finding study in monkeys indicated that exposure increased in a less than dose proportional manner with increasing doses from 30 mg per kg to 300 mg per kg. This was noted especially at the higher end of the dose range. Exposure in monkeys was similar to that achieved with the original DMSO containing formulation and sufficient to achieve the objectives of the tox studies. I'm now going to hand over to Mike, who will talk about translating a preclinical formulation to first-in-human studies.
Thanks, Alison, and good afternoon, everyone. For the next part of the session, I'd like to cover the considerations for translation from a preclinical formulation into a final formulation for oral delivery for first-in-human studies. And with that, I'm intending to cover the importance of defining a clear pathway for formulation selection, the importance of appropriate excipient selection and ensuring excipient compatibility early, a very top-level overview of formulation approaches that we can consider and the various pros and cons for each approach, and how we accommodate some of those processes here at Quay. Finally, to conclude with a case study covering a recent program that we've taken into clinic in order to give an indication of timelines and the activities involved. Firstly, it's important to stress that successful projects are based on sound scientific rationale and an understanding of the challenges that need to be overcome when converting the drug substance or API into the drug product. So the handover of information from drug discovery and preclinical studies is really important to be done to allow us to ensure that we have a good understanding of the physicochemical properties of the API and that this is well understood. No two molecules are the same, and our rationale at Quay is therefore to use the physicochemical characteristics of the API to drive the selection of the most appropriate formulation approach. To this effect, we don't offer any platform technologies, but endeavour to develop the most appropriate formulation for the challenge that's posed to us. If we understand the physicochemical properties of the API, such as solubility, the partition coefficient, and we have an understanding of the likely degradation mechanisms, then combined with an understanding of the permeability profile of the API and the pharmacokinetic data that's been generated during preclinical studies, then we can really start to narrow the scope of the options for evaluation during formulation development. But before we think about our formulation route, we need to define our problem statement. What is it exactly that we want to achieve? What does our target product profile look like? I'm sure that most people are familiar with the target product profile concept, so I won't labour the detail. However, for lipid formulations, I think that there are a couple of key factors that need to be clear before embarking on formulation design and ensuring that a lipid route is appropriate. Dose limitations tend to be the biggest challenge with lipid-based formulations. And also, lipid formulations tend to allow the inclusion of penetration enhancers more than most other formulation types. Therefore, it's useful to consider the implications of excipient selection early on and whether other APIs that may be co-administered with the API could possibly be affected in terms of bioavailability. Another key factor is that of the potency of the API indicated by its PDE value. A real benefit of lipid formulations is the potential to encapsulate highly potent APIs and reduce operator exposure risks, and this can really simplify production activities. If we understand our physicochemical properties and our target product profile, then we can really start to move forward to select our formulation approach. To help focus our formulation approach, we need to select the most appropriate excipients. And in order to do that, we need to establish two factors. Firstly, what function do we need the excipient to impart? And that could be the enhancement of solubility, to stabilise micellar systems, or to enhance permeation. And secondly then, is that excipient compatible chemically with the API? And as Alison mentioned earlier, lipid formulations may allow the opportunity to consider the incorporation of excipients that may improve bioavailability through their ability to inhibit efflux mechanisms. And this slide shows a nice representation taken from a paper published recently by a group from Liverpool University. And it identifies a range of excipients and ranks their effectiveness in increasing digoxin absorption. And digoxin is a model drug known to be an efflux substrate. And we can use this knowledge, therefore, to aid our selection of excipients and maximise the potential for absorption. To establish chemical compatibility, we need a stability indicating chromatographic method. And one of the problems with lipid-based excipients tends to be the potential of chromatographic interference of the excipients due to the tendency to absorb at low wavelengths. And we have a lot of experience in developing robust extraction techniques, including liquid-liquid and solid phase extraction, and this allows us to clean up samples where possible prior to injection. It's important, however, though, that we do have fully stability indicating chromatographic conditions, and that allows us to build a sound foundation for decision making about product stability. And the next couple of slides show some recent studies where we've had to optimize the gradient conditions to prevent interference of excipient related peaks with known API degradation markers. And as you can see on this chromatogram, we have a number of PEG-related peaks that are interfering with API oxidation markers. And this slide shows the chromatography following gradient optimization. And this allowed the interference to be reduced to that of baseline noise without influencing method sensitivity in relation to the oxidation marker. And on this slide it can be seen that there was a significant amount of carryover where the excipient was vitamin E TPGS, and this interfered with photolytic degradation peaks. So to improve the chromatography, high organic hold was employed to separate out the TPGS portion and separate this from the area of interest. We also incorporated a high flow rate in order to ensure that the method run times were manageable and that the TPGS could be removed from the column quite easily. So once we have confidence in excipient choices and a sound analytical basis upon which we can move forward, then we can progress into formulation development. Lipid formulations potentially can be used to produce a wide range of dosage forms, ranging from simple liquids through to more complex LIQUISOLID tablets. So if we consider liquid formulations initially, typically the formulation of choice for preclinical studies, so we may well have a starting point for our formulation based upon that used during preclinical evaluation. I don't intend to cover the mechanics of composition, as Alison previously described the solubility requirements and assessment process during her earlier slides. Some important things to consider though from an excipient perspective include that of the maximum daily intake values associated with the excipients, and this is of particular concern when developing pediatric formulations. The influence of excipient selection and concentration upon digestibility and droplet stability upon dispersion in biorelevant media is of particular importance when screening between formulations to establish leads. In addition to those functional excipients to maximize solubility and absorption potential, liquid-based formulations offer the potential for modification to improve patient compliance. And that could be through modification of flavor and mouthfeel enhancement using viscosity modifiers. Despite being considered as oily materials, some medium-chain triglycerides are actually quite palatable, and they don't require much improvement. But I think both me and Alison can confirm through our experience that a number of excipients do taste awful and require significant work to improve taste. Antioxidants may also be necessary, subject to interaction with various excipients in the API, and a number of excipients may contain residual peroxide as a result of the manufacturing process. And this is important to consider when selecting excipient grades and also the order of material addition when producing a formulation. The benefits of liquid lipid formulations are that of a very simple formulation route with possible rapid development times for first-in-human studies, a very simple process to scale and manufacture, and potentially dose-ranging studies can be used with a single formulation subject to acceptability of excipients. It really does allow the potential for rapid translation from preclinical into first-in-human. The second likely route of administration for liquid formulations is that of capsule delivery by either soft gel or hard gelatin or HPMC capsules. I'm going to focus on hard shell considerations, as that's the capability that we offer at Quay, and I believe the most popular route for oral delivery, with more formulation options for approach compared to soft gel. Irrespective of soft or hard gel approach, physical stability of the liquid formulation and the capsule shell is essential. There are known interactions between various excipients which cause either dehydration of the shell or interact with the shell, causing cross-linking, and this can influence the disintegration and dissolution properties of the drug product on stability. This slide shows a couple of examples of where incorrect excipient selection can cause some major problems. On the left, we have a HPMC capsule filled with caprylic acid, and you can see a large bulge in the capsule body. And this is where the caprylic acid has reacted with the HPMC, causing deformation. On the right-hand side, again, HPMC capsule with PEG 400, and you can see a significant crack right up the body of the shell. Now, these deformations were noted within a couple of days following introduction of the excipient into the capsule shells, and it really shows that incorrect excipient selection can have some drastic impacts upon the physical stability of the product. For liquid fill hard shell capsules, one of the biggest concerns with this type of product is that of capsule leakage. Particularly for early stage studies where products tend to be packed into pots, a single leaking capsule can spoil a bulk pot. At Quay, we look to band our capsules to ensure that no leakage takes place during stability or product storage. And you can see from the pictures that some capsules can exhibit quite large tolerances between the cap and the body, and therefore it's important that the banding process is able to overcome such intercapsule variability. The selection of band is specific to the selected capsule composition. Next, I'd like to consider semi-solid and capsule formulations. These tend to be type 4 formulations using materials such as high molecular weight PEGs, vitamin E TPGS, or the Gelucea range of materials. The bulk carriers are heated to around 60 degrees to produce liquids into which the API is then either dissolved or suspended. And the advantages of these types of formulations are that it's a very simple process. The filled capsules have a reduced potential for leakage as the material solidifies on cooling. There's the potential for higher drug loading when delivering suspensions compared to liquid formulations, and there's the potential for improved product stability. Over the past 18 years at Quay, we've worked on a lot of these type 4 formulations, particularly in the oncology space, and some of which have subsequently been commercialized. It's possible to convert both liquid and semi-solid formulations into dry powder products through the use of suitable adsorbent materials to produce LIQUISOLID formulations. These adsorbent materials, such as the mesoporous silica-based excipients, can absorb a significant amount of liquid whilst remaining as a free-flowing powder. The figure shows Nucilin US2, which is an alumino-magnesium silicate from Fuji Chemicals, loaded with Labrafac lipophile from GAPFSA. And it can be seen that the material can hold up to 1.7 times its own weight whilst remaining as a free-flowing powder. This figure shows the increase in bulk and tap density of the loaded material, which is important for the understanding of fill weight that can be achieved when looking to fill the powders directly into capsules. For the next couple of slides, I'd like to show you how simple it is to convert the liquid lipid formulation into a free-flowing powder using the adsorbent Nucilin US2. In these examples, we've used the SMEDS pre-concentrate formulation, a type 3 lipid product, containing dipyridamole as the API. Dipyridamole is ideal for this demonstration as it has a lovely yellow fluorescent colour, so it can show how easily the liquid formulation is absorbed by the Nucilin granules. The liquid formulation was simply added from the syringe dropwise over around 2 minutes, and you can see that the uniform distribution was achieved easily using a simple overhead stirrer without a complicated mixing process. For these samples, the powder was loaded with 9 parts liquid to 10 parts powder, 90% loaded. And finally, as you can see, the resultant loaded granules are so freely flowing that it's difficult to pour them into the bulk container without spilling it all over the bench. The data presented here confirms what we've just seen. The flow properties of the 90% loaded material and other variants that were loaded to a lesser degree at 50 and 70% are excellent and ideal for capsule filling. So why would we consider converting liquids into powder-based dosage forms? Well, there are a number of advantages associated with powders over liquids. Powders can be filled using standard solid dosage equipment, and the benefits over liquid capsules include less potential for interaction with the capsule shells. It may be possible to produce combination products where compatibility issues may be reduced through lipid encapsulation. We can use it to improve the distribution or content uniformity of low-dose products, and there shouldn't be a need to band or seal capsule shells to prevent leakage. There are, however, some limitations. API loading capacity could be limited due to the dissolution capacity of the lipid formulation, or the adsorbent capacity could be limited to the lipid formulation characteristics and the physical properties of the loaded granules. We also need to ensure that we've established the release characteristics of the API from the adsorbent in order to ensure that we get complete API release during in vitro and in vivo studies. It's possible to take loaded powders one step further and to convert these into tablet formulations. There are a number of critical factors that need to be considered though during development of these tablet formulations. Firstly, the adsorbent selection, and in our experience it's probably the alumino-metallosilicates that are the best absorbents for this type of formulation work. The level at which lipids are loaded onto the adsorbent are really key for tablet characteristics, and as mentioned earlier, the affinity of the API for the adsorbent in terms of in vitro release. We need to be very careful when we select our compression parameters, and press speed and applied force are really likely to be critical. And there really is a critical point at which the liquid phase predominates and this really is key to ensuring that new bond formation is created during compression. For the next few slides, I'd like to show you some data generated on a range of liquid-solid tablet formulations. A SMEDS pre-concentrate was loaded onto the adsorbent at 50, 70 and 90% loading levels. Extragranular excipients, mannitol, calcium carbonate and microcrystalline cellulose were added at 10% weight over weight extragranularly, and this was to establish the influence of extragranular excipients upon the tablet characteristics. A range of compression forces were applied to establish the influence of force upon the tablet characteristics also. The data shows that tablet robustness, tensile strength, is directly linked to loading concentration. We could see that as the liquid content was increased, the tensile strength was reduced. Viable tablets, and that's tablets above 1 megapascal, were produced up to 70% loading, and at 70% loading then, the viability of tablets appeared to reduce. From a disintegration perspective, we can see that rapid disintegration times can easily be achieved, but with this pre-concentrate formulation, we can see that disintegration time increases with increasing force applied and the pre-concentrate load. In this series of studies, it was shown that the most suitable extragranular excipient was microcrystalline cellulose to produce tablets with relatively high tensile strength and suitably low disintegration times. Tablet characteristics are influenced by the composition of the substrate loaded. The next slides show tablet characteristics for a range of formulations containing semi-solids, type 4 lipids, where the substrate loaded was either molten jelly CER 4414 or molten vitamin E TPGS containing dipyridamole. We can see that when compared to the previous formulations, suitably robust tablets, greater than 1 megapascal tensile strength, can be produced with up to 90% liquid loading and without extragranular excipient addition, therefore increasing the potential for greater API loading per unit. The friability of these tablets was also very low. The disintegration time for the tablets shows a linear increase with loading level and applied force. This is particularly the case for the loaded vitamin E TPGS samples, although all tablets still remain below 10 minutes for disintegration time. There are a number of studies published which suggest that one of the major concerns regarding liquid-solid dosage forms is recovery of API from the adsorbent matrix during in vitro dissolution assessment. These figures show that the excipient selection and process parameters are likely to influence the rate and extent of in vitro dissolution. But here we can see that a robust dipyridamole jelly CER-based formulation can be achieved, which allows rapid and complete dissolution irrespective of the compression force applied. So we've covered a range of formulation types that could be considered for phase 1 studies. Once we've characterised our formulation and have assured the stability of the drug product, then we need to consider how to scale the process and progress for clinical batch production. I'm going to focus on liquid-filled capsules for the rest of the session, as in our experience this tends to be the most favoured form for first-in-human studies. The process involves 4 main stages, including dispensing and mixing, capsule filling and check weighing, banding and then primary packaging. This is far fewer steps than other solid dosage form processing. For first-in-human studies, the scale of operation can be variable subject to the indication or the trial aims. The volumes for clinic may be outnumbered by the number of units required for QC retains, release testing, or to be tested throughout the stability program. A manual approach for capsule filling may therefore be more appropriate than scaling for automation, and this is probably based in terms of API usage and speed into batch manufacture. At Quay, we're very flexible in our approach and help to drive project timelines. Other considerations may be the environmental controls necessary to either protect the operators during manufacture or the API during processing. The video here shows a recent campaign where to protect the API we were required to manufacture under both yellow light and under nitrogen. To do this, we employ a range of engineering solutions within our GMP rooms to accommodate the process requirements. Some of these may be bespoke according to the project that we're working on. Where first-in-man studies are required to produce larger volumes to support trials, automation of the process can be beneficial to reduce manufacturing times. The video here shows our early phase automated filling capability. And these units allow us to produce tens of thousand units per shift. Due to the small footprint of these units, it's also possible to isolate these machines when necessary, as we saw in the earlier video. And this gives us maximum flexibility as we start to scale the process and allows suitable control from the environmental conditions and also isolation for the operators. This slide provides an overview of our banding process. All of our banding processes are performed automatically, and these pictures show the process from machine loading, top left, moving round clockwise to orientation on the machine, band application, and then drying prior to ejection from the machine fully banded. Selection of primary packaging components is an important consideration for dosage form design and for trial design too. A lot of early phase products tend to be filled into HDPE containers. At Quay, we also have the capability to blister pack and wallet for early phase studies, and we're also able to offer design services through our sister company, Chester Medical Solutions. Blister packs may offer improved protection where necessary, and we're able to provide both thermoform and cold form options. So hopefully that's provided a bit of an overview of the considerations that need to be made when taking a drug substance through from preclinical studies in order to establish a formulation that can be taken through into first-in-man. I thought it'd be useful to finish on a case study and to provide an example of a project that we've recently taken through and released for clinic. So for this particular case study, the drug substance was classed as a BCS2 candidate. From the preclinical studies and the pre-formulation studies, it was established that the solubility was approximately 0.1 mg per mil. The API was permeable, but it was shown that there was significant inter and intra-subject variability during animal PK studies. The log P for the compound was greater than 4, and a relatively large dose range was to be achieved in the first-in-man studies from 1 mg to up to 100 mg. And the client really wanted to be aggressive in order to get the drug product into clinic very quickly. For this product then, a type 4 lipid formulation was selected, and this allowed an improved dissolution rate and improved the wetting capability of the API. An excipient was selected known to inhibit efflux mechanisms, and that was vitamin E TPGS, and this allowed also the potential to reduce aggregation in vivo. The production process was a very simple two-stage process, and the PDE value was relatively high for the API, and this allowed us to contain the API in the molten lipid, which allowed then a very simple process, easy to scale in production. Firstly, apologies that this is quite a busy slide. What this shows us is an overview of the activities involved from setting the target product profile through to ICH stability studies on the clinical drug product. In order to hit the aggressive timelines that the client wanted to achieve, we ran a number of project stages in parallel, including excipient compatibility and formulation development studies. This required us to hold a number of key data review points to ensure that we could mitigate project risk throughout the project and ensure that we had a sound basis upon which to drive decision-making aspects. The total duration for the project was just over 5 months, from setting the target product profile through to release of the clinical drug product. Hopefully that's provided an overview of considerations for developing lipid products for moving into first-in-man studies. Before we move on to questions, let me give you an overview of Quay Pharma.
Quay Pharma is an independent, privately owned contract development manufacturing organisation. Since 2002, we have proudly delivered over 250 successful projects and exercised continued improvement whilst remaining true to our core values. Our use of proven manufacturing technologies provides a low economic risk and fast entry into first-in-human trials and accelerated scale-up to future clinical phases. We have knowledge and expertise in the formulation and development of a wide range of product formats and dosage forms for modified and targeted release within the body. We are licensed for the clinical and commercial manufacturing of new chemical entities, biologicals and live biotherapeutics in a non-sterile environment. The core strength of Quay Pharma is our people. We invest in the best. Only experts with strong scientific backgrounds. Their years of extensive knowledge underpins the service we deliver. We are highly experienced in employing process technologies to formulate and analyse new chemical entities that prevent particular challenges in solubility and permeability. We have highly experienced analytical specialists working alongside our formulation and manufacturing teams who provide expert advice and an enormous breadth of advanced analytical capabilities. Quay Pharma's flexible approach allows us to provide our customers with bespoke solutions to ensure on-time clinical delivery. Quay Pharma is committed to finding the best formulation and dosage delivery solution for every product, whether it is a new chemical entity, biological, or a live biotherapeutic, to give it the best possible chances of clinical and commercial success. We continually provide the highest quality in every aspect of the work we do. Our experienced team at Quay Pharma will rapidly screen and provide optimised formulations ahead of preclinical studies. Information learned in these studies will allow us to quickly move into clinical formulation stages, saving time on your overall clinical plan. Our innovative scientific approach is supported by our commitment to investment. This is clearly demonstrated at our growing facilities and will continue to be critical to our service offering. We innovate and challenge ourselves to be the very best in our field. Our aim is to continually develop pharmaceutical products with world-leading expertise that support safety, quality and commercial viability. Contact Quay Pharma and let's talk project success.